Hey everyone, this is Daniel Vanselow and my infant strapped to me currently. I am a programmer, web developer, and visualization specialist working mostly in Dr. Keith Chang's lab, but I also do some work for Dr. Yongsoo Kim. So if you've ever seen this beautiful fish rendering or this other mouse brain, that was me. So I normally do web development trying to visualize these very large two-dimensional data sets. They can have Z-series like this one, or they can just be huge whole-slide pictures. So I work trying to enable access to these as normal people without specialized hardware. So I also do sort of these advanced overlay data sets. Here's an anatomical atlas for a mouse brain that we worked on last year. But today I'll be walking you guys through 3d.fish, which is a web tool that we developed to share our very large micro CT 3D data sets, which brings us to the actual talk, which is 3d.fish, a cell resolution digital zebrafish. And there's a lovely subtitle, which is web-enabled data sharing and visualization for next generation image modalities. In order to get to the web tool, I'm going to very briefly walk everyone through what micro CT actually is, what the raw data looks like, and then why we might need to do something special to get it into a format that we can actually see on the web. Micro CT is an X-ray based imaging modality. That generally means for our sort of samples, you need to stain with a heavy metal, and that heavy metal will attenuate X-rays and generate signal. After embedding our samples in a rigid acrylic resin, we put them in the path of an X-ray beam and image the sample by rotating it around 180 degrees, and we take an image at each fraction of a degree to generate 1501 projections or images. We then restructure the data in this sort of third dimension, which is normally depth, but for us it's angular, so it's an angular dimension, or maybe a time dimension, since we're rotating the sample about an axis over time. And this domain is called sinograms. So if you take a look at this cube, as we raise the image, each of these pictures you see are sinograms. And we reconstruct the digital zebrafish to the right here by performing the radon transformation on each one of those sinograms. I'm going to show you some of our actual data from the setup at Argonne National Labs. This is a 5-day post-fertilization zebrafish. I'm scrolling through the stack from angle 0 to angle 180, so image 0 to image 1501. As you see, the fish is rotating around an axis, and when I get to the end, it should be a mirror image of the first image, 180 degrees later. So if we sort of look through this stack of images horizontally, if we take one slice through that angular dimension, we can go and see the corresponding sinogram, which is about slice 300 or so. So if you look at the sinogram, you can kind of see those lenses moving throughout space. It looks messy, but if you perform the radon transformation on each one of those sinograms, you get these really beautiful cellular resolution images. And if you combine those together, you get the full digitized zebrafish. So we're currently looking at the head of the zebrafish, and as I traverse down the stack, you can see the gut and swim bladder. Now we're back at the head, and you can see the lenses, which are those white circles in the brain of the zebrafish. If you shift your attention to the top left of this current window, you'll see that the image stack is actually 32 gigabytes, which is just totally massive if you want someone to download your file to look at it. Just keep that in mind. Now that we're halfway through the presentation and still haven't gotten to the web app, one last distraction. As an example of what you can do visually with this data, this is a 33-day post-fertilization zebrafish using the same method. These are the gills, and we're sort of cutting away the fish with 2 cut planes, and you can see sort of how beautiful this data can be if you treat it correctly. Dr. Chang loves to say that these bumps you see on the gills are the individual cells that are captured with this method visually. They're kind of like sweeping underneath and through the gills. This is one of our favorite videos. details. So our dream here is to take these really large data sets, which easily run over 100 gigabytes, and make them available to anyone. So like the public or researchers. We want to be able to sort of image anything and throw it online with zero barrier of entry. And that's sort of how we approached 3d.fish. So I'll just walk through the use of 3d.fish, how to load it, what it does. I'll type in the address 3d.fish. It'll load a bunch of images. It's already done. That was really fast. So here we're met with that 5-day post-fertilization zebrafish. It's laid out in multiplanar view, which is what radiologists typically use to look at your CT scans. So you can scroll through each orthogonal view independently. Here we're scrolling through transverse. We just scroll through sagittal. You see these yellow, blue, and red cut planes, and they all correspond to each other in space. They're all linked because the data is isotropic, meaning that X, Y, and Z are all the same dimensionally speaking. So these are all 0.743 microns. You can use your mouse and sort of pull around where the viewer is looking, and it will pull up the corresponding images. So I'm just clicking through the included functions, which includes inverting the color space and zoom. So if you click zoom, it will load in a high resolution image of whatever cut plane you'd last clicked on. So by default, in that multiplanar view, it loads in some very, very downsampled images and about a fifth of the data set total. Here we're looking at slices in this series of the sagittal section of the zebrafish. And we can scroll through those slices available to us and then click one and it'll go to that slice. So here we can see the eye of the zebrafish. We'll go back to the middle. You can go single slice by single slice. And we've also included the ability to do rudimentary labels. So I can pull up a rectangle and select something of interest. Let's say, if I can spell and type correctly, the pneumatic duct. And we can hit OK and initiate that label. And say we want another label, say the brain, and maybe also the heart. Let's see if there's something else we want to label. Maybe, let's look. A section of motor neurons, perhaps. And then what we can do if we want to ask our PI what these things are or confirm something or just share it with someone who might be interested is you can generate the label, copy the label, enter the label into a new address bar, and the labels are there. So if you click the label button, it has a pull down with all of those structures that you labeled. And it'll automatically pull this window up. You don't have to click on zoom. So this is for just some rudimentary interrogation of the data with maybe someone else, or you're teaching a lecture and you want to label some anatomical features. So the function's also built in with this initial multiplanar view where you click on the link generator, generate a link. You can send just those particular coordinates in your orthogonal fish. You don't have to be in the zoom. You don't have to generate labels. The current iteration of 3d.fish has a number of samples. We're looking at a 5-day post-fertilization fish, but we have a 33-day, 2, 3, 4, 5-day post-fertilization of wild type and our mutant Hulihutu. Right now I'm loading a 2-day post-fertilization fish. And you can see this one's a bit smaller and less developed. So you can do the same things we did with that 5-day fish, zoom in, interrogate at a higher resolution. I'm going to load that 33-day sample that you saw the video of earlier when it dove into the gills with the 2 cut planes pulling apart. This one takes a little bit longer to load because the images are a bit bigger, but we're talking about 13 megabytes per sample if they're large, which is a far more realistic scenario than the 32 gigabytes. This fish itself was about 100 gigabytes before we put it in here, and I believe it is 15 megabytes to sort of initiate this viewer. So we're going in and we're going to look at these same gills that we made the video of earlier. And just going to look at the data. It's no hands. It's really beautiful. And you can go here. It's the address is 3d.fish, and load these up and look at them anytime. You can send us links to it. This is one of the fun features we have where it projects the data, the number of slices you choose, and it makes a maximum intensity projection image that you can then zoom in and look at. I'll do it with a couple of the cut planes here. There's some options in there if you want to explore what it really means to be a maximum intensity projection. But it's basically taking the brightest pixel at each coordinate and keeping that pixel, making a projection image and keeping the maximum intensity. So you can kind of see things like vessels very well, or nerve tracks, if this was not a PTA stained fish. All these fish in this viewer are stained with PTA. But we have a few with uranium osmium. We have some unsand samples that we haven't put up yet. But some really beautiful data, and it's really easy to get to and really easy to access. We spent a lot of time sort of getting to this point. Along with a totally revamped labeling system, one of our goals in the very near future is to incorporate an actual 3D component to this viewer. We figured out how to really aggressively downsample our full image stack and interrogate it in a three-dimensional viewer. And so our goal is to take this sample and stick it in the corner there for a better idea of where you are in three-dimensional space in your orthogonal cut plane views, because it's kind of hard to know, unless you're an expert, where exactly you are in each stack of each sample. So our idea is to grab this very downsampled little fish and throw them in the corner and sort of have each cut plane represented on the sample itself in 3 dimensions. That's it. I'd like to thank everyone from Keats Lab and the CT physicist collaborators we have. They're amazing. And NIH for funding us to do this fun stuff, as well, of course, as the Zebrafish Functional Genomics Core, because without them, we would not have any of this. Thanks, guys. Questions?